So I do have one slide of background, and this is just to reiterate the importance of the C9ORF genetic anomaly in patients with ALS and FTD. The numbers may not be up to date, and also they may not include China or the Asia continent for that matter, but it's striking how the frequency of the pathogenic GGGGCC repeat expansion in C9ORF is high in sporadic ALS cases, up to about 6%, and especially in the familial cases of ALS, up to 40%, as well as in the sporadic and familial cases of frontotemporal dementia. And the frequency goes as high as 80% in patients that have both ALS and FTD. So it's a very, very important gene. And in the context of familial ALS cases, the C9ORF mutation is the most represented. You can see it here. It would be probably unlikely that we will have another gene that can cover the remainder of all the cases. It will be a number of genes that represent a very small population. So this is a very important gene. So what we know about the function and localization of the C9ORF protein is very little or none. The function and localization remain completely uncharacterized. This is just a background slide to say that we have 3 transcripts that encode up to two isoform A, which is the longest one, and isoform B. And I would say that the study on the C9ORF protein has been hampered by the lack of a fully characterized antibody that probably put a problem there. So we did a little bit of characterization there using shRNA approach. You can see here we transfected our construct. These are NSC34 cells. And you can see here that our shRNA is able to knock down, almost out, the totality of the C9ORF isoform A here. And then when we transfect NSC34 cells with the isoform A and isoform B, our antibody that we had in the lab is able to pick up the 2 isoform, transfected isoform with equal affinity. So as well as the A and B are equally represented on this plot. And you can see from the control that our antibody is able to pick up the isoform A. This is a flag isoform, so it's slightly higher. But our antibody can pick up the isoform A, but not the isoform B. So most likely in these cells, the ratio between the isoforms is slightly different. So we took this construct and we put it into the cells and look at the immunocytochemistry. And this is the results with the antibody. You can see that both isoform A and isoform B have a prevalent expression in the cytosol. And also you can see that the antibody is not able to pick up the endogenous isoform, so suggesting that the endogenous C9ORF is very low in these proteins. Those were NSC34 cells. These are cortical neurons, so we look into cortical neurons. This is a group of cortical neurons that were transfected with our construct, which is tagged with the GFP, shRNA construct, which is tagged with the GFP, and show you that one of those neurons got the transcript, the shRNA. And you can see that the antibody recognizes a cytosolic localization, cytosolic C9ORF protein. This is well shown here. When you look at the neuron that has the shRNA, you can see the cytosolic staining completely disappears. So we are fairly confident that in neurons, the C9ORF protein is localized in the cytosols. And also, differently from the NSC34 cells, seems like the neurons express higher level of C9ORF, at least judging from this immunocytochemistry data. So going back to the pathogenicity of the C9ORF repeat expansion, I guess the $1 million question, maybe more than $1 million. $1 million is just an NIH grant, so maybe more than that, is what are the C9ORF pathogenic determinants, and most of all, what are the mechanisms that mediate C9ORF toxicity. So we heard a lot about the possible hypothesis. We have a loss of function of the C9ORF protein, and this is justified by the fact that there is a decrease in the reduction of C9ORF transcript in these patients. So the loss of function hypothesis was put forward. There's some data out there suggesting that knocking down C9ORF could be toxic, but also we heard about today that that's probably not the case. We have data, which I'm not going to talk about, using our shRNA, that if we knock it down from neurons in culture, the neurons are perfectly fine. So we tend to support the hypothesis that C9ORF haploinsufficiency is not a cause of disease. On the other hand, we have a gain-of-function hypothesis of the C9ORF transcript, and we heard about today a lot about the G quadruplex formation being toxic, or the RNA foci that binds RNA binding protein being toxic. I'm not talking about this also because I don't have much of data to talk about. What I'm going to focus is the hypothesis that these translated peptides can be toxic, could be toxic, and we are going to show you. I'm going to test the hypothesis on in vitro culture models, and also I'm going to show you a little bit at the end of the talk in vivo modeling, suggesting that some of these dipeptide proteins are indeed neurotoxic. So we used a strategy to generate different construct that encodes these C9ORF RAN-translated proteins. We use, as well as Stuart showed before, a randomized codon strategy to avoid or separate the toxicity of the RNA transcript, avoid high GC contact, which could destabilize the transcript first, but also avoid the potential intrinsic toxicity of a long G4C2 repeat sequence in the transcript. So we want to make sure that that was not, what we see was not related to the RNA transcript. And with this strategy in our hand, we were able to generate different, all 5 potential generated dipeptide at different lengths, and this is the lengths that we were able to have from 25 to 400 for GA and GR, and from 25 to 200 for PA and PR. I'm talking about stable in our hands. We were not able to go higher than 50 for GP for the problem that the constant, if you go higher, we had a lot of G and C in the sequence, and that would destabilize or impair our ability to amplify the construct. So this is what we're going to test. So with this construct, we did some preliminary experiment to look at the localization and possibly aggregation pattern of these C9 polypeptides. We used, again, the NSC34 cell lines. These are straight Western blot from this cell transfected with the construct using the antibody that were graciously provided to us by Len Petruccelli. So you can see that the constructs are equally expressed in these cell lines, and these are probe for the GFP tag. I forgot to say that our constructs are GFP and flag tags, so we can use those tags to monitor the expression and localization levels of this transcript. So if you go through the panels here, you will see that the GFP obviously is not giving any interesting localization pattern, remaining diffuse in the cells. GA, we tested GA50, but what I'm going to tell you here is the same for all the construct at different lengths. So this is a representative length that we use throughout the talk, that I'm going to use throughout the talk. But what we see here is also true for the other lengths. So GA tend to localize, aggregate, quite strikingly in the cytosols, maybe a little bit perinuclear, especially with the IL length, but remaining in the cytosols as an aggregate. GP50 remain diffuse throughout the cells. GR50 striking a nuclear localization. PA is somehow in aggregated form, shown here, but mainly diffuse. I'm going to show you some data of light cell imaging showing that PA can aggregate but also disaggregate. So it's a dynamic situation. PR50 instead is strikingly nuclear localization, similar to GR. So this is the situation in the NSC34 cells. And so we took the next step and we moved to primary neuron, looking at their localization with our construct in 2 neuronal type that are actually relevant to the disease. So we use primary cortical neurons and primary motor neurons as well from rats. And this is just an example of our transfection protocols. We use a co-transfection strategy. We use our C9 construct, they are GFP tag, but along with those, we put also the TD tomato construct driven by the synapsin promoter, so we can actually do some nice tracking of the neurons, and as well as some live cell imaging. I'm going to show you the data a little later. Sorry. The ratio between the construct is 4 to 1 4 being the C9ORF, and the C9 construct to 1. So just to make sure that all the cells that carry the TD tomato also carry the our construct. These are the first localization data. You can see that GA50 localize form cytosolic aggregates in the cell body, but also in the axon and then possibly dendrite. There's some also in the dendrite. So it's pretty striking localization there. Time-lapse images show these aggregates endure over time. Hopefully you can see it here that these are 3 neurons that have been transfected at day 0. When we do transfection at day 0 means that they are transfected at day 10 in vitro, so the neurons are already fully mature. What you see here is just the expression pattern of the synapsin promoter that drives the expression of the TD tomato. So these are already fully mature neurons. But you can see that these 3 neurons carries the aggregate, and you can see that aggregate persists over time, up to 7 days, 8 days. That's why we censor the data. And also maybe you can also infer from this data that GA in our hands doesn't seem to be toxic. This is the data for GP50 and PA50, and you can see that PA50 and GP50 has cytosolic localization, diffuse expression pattern in the cortical neurons. As I was mentioning before, that sometimes PA aggregates, but the aggregates are dynamic. The situation is shown here. If you focus on this neuron, you will see that PA seems to aggregate, and this is the day 2. But when you switch and look at the same neuron at day 5, you will see that the aggregate disappear, remaining a diffuse localization. So it's sort of a dynamic situation of the PA aggregates. These are the data for PR and GR50. So GR50 is as a nuclear localization, but as well as a strong localization in the cytosols. This is a TD tomato positive neuron, so you can see it here, GR50 nuclear plus cytosol. However, PR50 has a very strong tendency to accumulate in the nucleus. There is no way we can see. There is some, I will show you data later, but mostly PR50 goes into the nucleus. This is just to show you that those were the tag version, these are untagged version. It's the same thing we do see with the antibody, the polyclonal antibody that recognize the peptides themselves. You can see the same aggregation or localization pattern throughout the D peptide. We'll switch to the mononeurons because those are the cells that are relevant to ALS, pure ALS. This is our protocol. We can show in here that we are able to culture pure mononeurons, P75 and CHAD positive mononeurons. And we did repeat the transfection, and we also saw the same thing here. So GA50 along the axon, and sometimes also dendrites, but sometimes in the cell body, GR50 nuclear plus cytosolic, PA diffuse, GP diffuse, PR50 strikingly in the nucleus. So, so now back to the $1 million or 1 NIH grant question. Are these dipeptide neurotoxics? And if they are neurotoxic, is the neurotoxicity selective to the neuronal type? Yeah. What's the tag? I'm sorry. Do you have the tag? Oh, the tag, a GFP tag. Yeah. So are these neurotoxicity neuron selective? And most of all, if they are neurotoxic, what is the mechanism? So to answer the first question, we turn to a time-lapse imaging experiment for assessment of the toxicity of this polypeptide. This is, again, a reminder of our protocol. These are cortical neurons. We transfect them at day 10 in vitro when they are already fully mature, and then we can follow throughout time with time-lapse imaging the population of neurons that were transfected and whether they die or not. We classify a dead model or a dead neuron as a neuron that disappeared from the imaging compared to the day before. We image these neurons every 24 hours. And being able to deal with a population of neurons, also you can apply Kaplan-Meier survival analysis and look at whether this neurons population die over time according to whether they are transfected or not. And this is a control situation. You can see that we can keep this cortical neuron for 8 days post-transfection fairly well. And then after 8 days, we censor the data because we have a dropout of about 20% in 8 days. So with this in mind, we put our construct into these neurons, and you can see it here, right here, focusing on this Kaplan-Meier survival curve, that cortical neurons are extremely sensitive to, or their survival rate is very low. If they are transfected with PR50, they drop out quite quickly, whereas the other dipeptide seems not to have any effect on these neurons. And this is shown here by these representative images. You can see a control. These neurons are transfected with the PR. You can see that they disappear quite quickly off of the imaging field. The situation seems to be a little bit more complicated than that, because it seems like the neurotoxicity correlate with the presence of aggregates. I would like to focus on this neuron here, day 1, that carries an aggregate of PR, and you can see that the following days the neuron is gone, whereas this neuron here didn't carry aggregate, but when it starts carrying aggregates, and so it's perfectly fine, when it starts having aggregates of PR, oops, sorry, what's happening? Oh, sorry. So I was saying this neuron here that seems perfectly fine at day 1, day 2, at day 3 start to have an aggregate of PR50, and then the following day is bound to die. I was mentioning aggregation versus not aggregation for PR. We have also neurons that didn't carry aggregates. This is a small percentage of this neuron, like less than 10% of this neuron have a diffuse pattern, and the ones that are able to keep PR in the diffuse state are the ones that actually survive. This neuron here is shown here as a diffuse PR staining, and it's perfectly fine. In fact, we can break down our survival analysis by the neurons that have diffuse aggregation versus the neurons that have aggregates, and you can see clearly here from the cumulative survival analysis that PR aggregation is the killing factor, whereas, and also this is a cumulative risk of death, whereas the diffuse aggregation or diffuse localization pattern is not toxic. We did some length-dependent toxicity, just as was shown before. PR is not, the PR toxicity is not dose-dependent. It's very toxic already at very short length, 25 throughout 200. This is, say, ANOVA. There is no difference in the dose dependency. GR is dose dependent. At 50, these are cortical neurons. At 50 is not toxic, but it starts to become toxic as you go high. The other one seems to have a trend in toxicity, although some toxicity was detected for GA at toxicity at lengths as high as 400, but it's minimal compared to what we see for these two polypeptides. And the other one didn't show, GP didn't show anything, but we couldn't go as higher than 50. Motor neurons, they behave the same. The toxicity of PR is similar to what we saw in the cortical neuron, but in this class of neurons, we also saw toxicity for GR50 compared to the others. The others are not toxic, but GR and PR50 are toxic, significantly toxic. And here it seems that the toxicity, again, seems to correlate with the presence of aggregate. And this is just a movie that we put together, a live cell imaging of a motor neuron carrying 2 tiny aggregates in the nucleus, presumably. And you will see that these 2 aggregates, as we image these neurons across time, this is about 8 hours imaging. So don't worry, I'm not going to keep you here for 8 hours. But what we're going to show you is these 2 aggregates converge into a larger aggregate, and as soon as that happens, which is probably within 3 or 4 hours, these neurons start to degenerate, starting from the neurites, which become fragmented, and the soma, which starts to swell and then disappear. Oh, no, I want to start the movie. If I can. Okay. All right, you can, okay, it just started. You can see that these 2 aggregates converge together and start to become a big one. And then if you keep an eye to the dendrite, as soon as this happens, which is probably around 3-hour time point, they start to fragment, degenerate, the soma start to swell, and the cell is bound to die. I just want to cut it short because there is an interesting, so the cell is dead at about 4 hours, and then it disappears. The interesting thing is that the aggregates stick there. It doesn't disappear as well. So it's very solid. It's like a rock sitting in the cell. It doesn't go away. The movie goes on for 8 hours, so it's still there. So that's a very interesting observation. Another thing that we notice using some FRAP analysis is that the PR aggregate, as remarkable as it is, they are dynamic. That means that they can reform quite quickly. You can see here from this movie, this is an aggregate of PR, nuclear aggregate of PR is photobleach. The fluorescence disappears, but then it recovers very quickly in a matter of a few seconds. That's quite remarkable. So you can see now it's photobleach, and then as time goes on, it comes back. So that's a very surprising observation. You wouldn't expect that from an aggregate, but that's the case. It doesn't recover as much as the full fluorescence that he had, but it comes back pretty quickly. This is instead is a GA aggregate, which is not nuclear, it's cytosolic. And you can actually see it from this graph profile here. It does not come back. So it's a very different kind of aggregate. You see it here. That was an interesting observation that brought us to imagine, this is not real science yet, but imagine a structural correlate toxicity. And if you run an algorithm to understand the structure of these aggregates, you can see that these are not, I'm sorry, these are not a GR as a type, as a GA aggregate. You can see that GA, they coil into a coherent structure with beta sheets and alpha helices, whereas the PR50 is not able to structure itself in any organized structure. It remains a coil or a twirling strip that doesn't aggregate or doesn't give a secondary structure. And this is shown here from a higher resolution of this aggregate. If you can run a find edges function on image J and look at this aggregate, you can see the striking difference between the two types of aggregates. PR are more diffuse, GA are more organized. So that was an interesting correlate that we may have. So we may have a lead there to understand the toxicity of these things. There is a neuron specificity in the toxicity of these aggregates. These are hippocampal neurons. They are still sensitive to PR, but they are much, much less sensitive to PR. The other ones are not toxic at all. And this is a combined graph between hippocampal neurons and cortical neurons. You can really see that the two type of neuron are sensitive, are differentially sensitive to the PR, much more the cortical neuron, much less the hippocampal neurons. So that was an interesting observation. So that was about toxicity, and now we try to dive into the possible mechanism. I show you that PR and GR aggregate in the nucleus, but where in the nucleus? And you can see it here, or probably you already read the title, that PR and also GR, I'll show you the data in the next slide, PR aggregates in the nucleolus, aggregates together with nucleolin. This is to confirm the data that Stuart showed before. You can see here this neuron TD tomato positive with the PR aggregates and stain for nucleolin. You can see that the PR forms these huge aggregates right there. And this is even shown even better. I agree with what Stuart was saying, that the PR sits in the middle and then you have these clumps of nucleolin around it, and this is nicely shown by this image. You can see it here. So it's a quite remarkable aggregation pattern that is very indicative of a mechanism of nucleolar stress response that occurs upon expression of these aggregates. These are the GR50 data. You can see that cytosolic localization, but if you slice through the cells, the cortical neuron, you would see the same aggregation pattern. Nucleolin, the GR aggregates in the middle, and nucleolin in the nucleolus aggregating around it. So similar to PR. We also observe that you have a dispersal of nucleolin, and this has happened, we happen to see it in the mononeurons. This is a PR expressing mononeurons. You can see here that nucleolin is dispersed. So it's another, again, sign of stress response, nuclear stress response. Another interesting thing that we notice is that this PR aggregation causes activation of several stress responses, including a decrease in the P-bodies. This is quite remarkable. You can see this. This is a control situation. You can see the number of cytosolic processing body that is normal. These are detected with a DCP1 antibody. But these are PR50. You can see that the number is slightly different, but also the size. So, and these are the data plotted here. We also were able to break it down by diffuse state versus aggregation state. And you can see that if you have aggregation, you have a remarkable reduction in these P-body numbers. That could mean is that you might have an increase in mRNA translation or decrease in mRNA silencing or an impaired granule assembly mechanism. All these hypotheses feed into the general hypothesis of a stress cell response. And this is just to confirm that we also stain for granule cells, stress granules markers, and you can see that PR expressing neurons, and these two neurons here, one is carry the PR's aggregation, and that one also have stress granules appearance, whereas the other one not. And this neuron here, if you stain it with MAP2, also show MAP2 disorganization. So this cell stress response correlate with toxicity as well. Another interesting phenomenon that we show is that the PR aggregates promote a near complete loss of nuclear fuss. And this is shown here by this example. These neurons, one of them have a PR50, and you can see that whereas the fuss, endogenous fuss, nicely localized and is restraining the nucleus, the PR50 neuron has a complete loss of nuclear fuss. It translocates into the cytosol, it aggregates there along the cytosol, along the cell body, the cell soma, but also in the neurites. So it's another clear sign of cell stress response. These are the GA data. The GA the GA aggregates don't do the same thing, although there's some mislocalization of the endogenous fuss that migrates along the neurites, but the majority of fuss remain into the cytosol, into the nucleus. So this is an extreme situation. We also were interested in looking at early sign of cell or neuronal impairment. And so we look at these neurons from their potential to release a neurotransmitter. This is a release assay. I just don't want to go into the detail of the assay, but early on during the transfection, if you look at this neuron and you measure their ability to unload synaptic vesicle, they are stained with a fluorescent dye, and you depolarize these neurons with high potassium, you can see here that the control nicely release, unload the vesicle. When the vesicle fuses, lose fluorescence, so that can be detected. But the PR and GR neurons lose that ability. This is plotted here as the slope of the initial decay rate of release. So it's quite clear that even at early stages, these neurons are somehow suffering. They basically die, and this is just a staining showing that when they are late in their neurodegeneration process, they show apoptosis. This is Kaspersky staining for a GR50 neurons expressing neurons. So based on this data, we tried to rescue this neuron, looking at whether blocking apoptosis or even blocking necroptosis was able to try to rescue this neuron. But if you do that individually, in other words, if you use a specific blocker of apoptosis, it doesn't do anything. The neuron is still dying. If you use a specific blocker of necroptosis, again, they don't die. If you put them together, there is some sort of rescue, but it's still not enough to rescue those neurons, meaning that these neurons are really suffering. And perhaps if you block one pathway, they can switch to another pathway. They find 1,000 ways to die. And so bottom line, we are not able to block these death pathways. In vitro data, these are in vivo data, just to show you that whether this aggregates or dipeptide were toxic in vivo. So we took the PR50 and PA and GA and we put it into the Drosophila using different drivers. You can see here, if you use the drive to drive the expression of this peptide in the eye only, there's a massive, massive degeneration of PR50 in the eye, and you see the eye phenotype is completely gone, whereas the PA50 and GA50 is normal. This is a plot of the eye degeneration. If you put those in the motor neurons, specifically using the motor neuron driver, the flies develop normally. This is a PR fly, develop normally up to the pupil stage. There's no eye degeneration. The only thing that is missing is the motor function. They are completely paralyzed and they don't make it out of the pupil case. They die immediately, so it's very little, although they develop normally. Motor neurons are gone. And in fact, going back to one of the questions, if you run a Western blot of this of the eye or the motor neuron here, you don't see the PR aggregates. It's very hard to see the PR aggregates. The only way to see these PR expressions or the other is to look at the neural tissue in the larvae. And this is, again, in staining, and this is just showing that it recapitulates the expression pattern that we observe in the culture neuron. So nuclear aggregation for PR, cytosolic for PA, and nuclear and cytosolic aggregation for GA. And this is when we express specifically our polypeptides in the muscles, we were able to detect them by Western blot. So we are pretty sure that proteins are made. The only thing that when the neurons degenerate, these things are invisible. So just to conclude here, what we want to propose here is that PR and to a lesser extent GR are the pathogenic RAM peptides. The other peptides, at least in our end, are mostly pathological hallmarks. The neurotoxicity of PR and GR correlates very well with their nuclear aggregation, and the mechanism of toxicity, we weren't able to pick up a specific mechanism. What we can say is that we have a massive global war going on that include transcription, translation, dysregulation. We have nuclear enlargement, stress granule formation, fast mislocalization, p-bodies decreased number. And also the data told us that blocking some of the pathway that can lead to a cell death, like apoptosis of necrosis, is no beneficial. Even if we put them together, you get some rescue, but not complete rescue. So there's a massive, I would call it transcriptomic crisis going on when you have a cell or a neuron making these PR or GR aggregates. And then I just want to conclude by proposing, these are two tables taken from published data from Land and from Laura Ranum, where they show formation of RAN inclusion. And what you see here, if you look at PR is not detected in the spinal cord as well as in here. That is the relevant area of neurodegeneration in these diseases. And that we are proposing that you don't see it because they are extremely toxic. Once they form, they kill, so the neuron disappear. And what you see here is what's left out. Certainly, if they have PR, they don't have it. You may see it in the hippocampus, but they show you hippocampus are less sensitive to PR, so they may find a way to manage those aggregates a little bit better than the relevant neuron in ALS FTD. And finally, I just want to thank one person in particular, a student, a PhD student of mine, who actually was at the center of this data generation. Xin Mei Wen is a very talented PhD student in the lab. I want to also thank collaboration with Uday and John Mohagam at UPIT, and obviously Len Petruccelli for the antibody sharing, and everybody else at the Weinberg Unit for ALS research. Thank you.
Thank you, Davide. Question?